Hello friends, I welcome you all. Today we will discuss some of the insights in the pathogenesis of viral diseases in animals. As we all know, animals can be affected by large number of infectious agents. Amongst many other infectious agents, viruses are of prime importance. Outcome of virus infection is essentially depend upon virulence of infecting virus as well as susceptibility of the host in which this virus causing infections. For virus to cause disease, they must first infect their host, spread within the host and damage the target tissues for which virus has got the tropism. To ensure their propagation, virus must be shared within secretions or excretion, so that virus can be taken up by vectors, or it can pass congenital to the fetus, or it may contaminate food and water so that it would transmit to another susceptible host and propagate. Viruses have developed remarkable variety of strategies to ensure their own survival. Similarly, to cause disease in a host, it has gained considerable variety of distinct pathogenic mechanisms. That mechanisms we are going to discuss in brief in this presentation. Merely viral infection is not a synonymous with a disease, or it is not considered as a disease, because many viral infections remains inapparent or asymptomatic, whereas some of the viral diseases leads to typical clinical disease with very typical clinical signs, as well as it may vary in severity, like it can be mild, moderate, severe, or some of the viral infections may lead to the death in a few animals as well. So to just cause the infection in a particular host, virus has to go through the various steps, and doing these, it has to evade the innate as well as adaptive immune response of that particular host. So to just cause the infection, it has to enter into the body, it has to spread locally as well as systematically. Afterwards, it has to shed into the secretions and excretions of that host, so that it can spread to the another susceptible host. By the time that particular host immune system will develop certain antibodies, adaptive immune response will come into play, and it can clear that virus and recovery can take place in that host. Sometimes it may happen that virus may remain persistent in that host, it can cause chronic infections, or it may lead to the death of that particular host. So to just enters into the susceptible host, virus has number of ways by which it can enters into that host. So most important route to enters into the host is the respiratory tract. Respiratory tract is a very important system of the body, and it has got the innate as well as adaptive immune mechanism that prevents the entry of such pathogenic virus inside the respiratory tract. Perhaps it is the most common portal of virus entry in that body of the animals. It is generally provided with the mucociliary blanket as well as alveolar macrophages, secretory antibodies, which prevents the attachment of these virus particles with the respiratory epithelium, so that it cannot cause the infections. A substantial number of viruses are also spread to susceptible hosts by ingestion of virus contaminated food and drinks through gastrointestinal tract. Similarly, another important organ, that is skin, it is the largest organ, and it is just covered with the thick layer of keratin, which prevents the entry of virus, but breach in the integrity of the skin by insect or animal bite, cuts, punctures, or abrasions predisposes to the viral infections. Another route of transmission includes venereal transmission, in which through natural coitus or through genital routes, virus can enter from infected to susceptible host. Through conjunctiva also, some of the viruses can enter into the host body, and it may cause the infections. So once this virus enters into the susceptible host, it has to spread in locally over the epithelial surfaces, as well as it has to invade the subepithelial tissues and reach to the lymph nodes, and then it has to enter into the blood. Through blood it will cause viremia and it will spread throughout the body. While doing all these, it has to interact with various components of that body, including monocytes, endothelial cells, as well as some of the viruses are there which uses the nerve to spread within the body, so that it can avoid the adaptive immune response or antibody produced against these bodies and clearance of this virus. So monocytes and macrophages are important component of innate immune system, and it helps to clear virus infection. Also it presents the viruses to the other immune component of the body. So it can elicit innate immune response by producing type 1 interferon or pro-inflammatory cytokines. At the same time, some viruses can maintain themselves in the cytoplasm of that particular macrophages or monocytes, and they keep on multiplying and produce new progeny, as well as these monocytes can act as a carrier for the virus to the various organs, and this is how virus can avoid the antibodies produced against it in the body. So this is this slides illustrates how this virus enters into the body, which cause primary viremia, secondary viremia, and then lesions, and how it spreads to the other susceptible host. So virus has to enters to the epithelial surfaces, in the subepithelial, and then to the subepithelial tissue. In the subepithelial tissues, these surveillance cells like dendritic cells or macrophages will be there. They will take this virus to the lymph nodes, and here primary multiplication will take place. After primary multiplication, small amount of virus will be released into the blood, that will reach to the organs of tropism, where there will be the secondary multiplication, and a large amount of virus will be released into the blood, that will cause secondary viremia, and it can reach to the various organs and can produce the damage and typical lesions, and then typical disease with clinical signs. At the same time, this virus can be available in the excretions, secretions, and in the body fluid to transfer it to the susceptible host. This is a very good narration of the ectromelia virus experimentally infected mouse, wherein at 0 day this virus was inoculated in the skin. Within a day it reached to regional lymph nodes, and afterwards into the blood. It causes primary viremia. Afterwards it has just reached to the organ of tropism, that is spleen and liver, and then multiplied, and then again secondary viremia, and afterwards by the day 10 it reaches to the skin and causes severe rashes and ulceration over the skin of that particular host. So just this virus, when it reaches to the susceptible host and invade the cell, there are particular steps in the replication of this virus. It has to attach to the host cell membrane, and this is facilitated by the host cell receptors and viral proteins. Then it will penetrate its genetic material into the cell cytoplasm. The genetic material will comes into the nucleus. There will be the formation of viral proteins, and then it will just assemble, and there will be just release of this virus particle. Viruses typically encodes the gene that modulate host cell function for their own benefits. So while doing all these, it will reduce the host protein synthesis, which are necessary for the maintenance of host cells. Then it will produce some of the metabolites that will be the toxic for these cells, so that subsequently will lead to the lysis and necrosis and degeneration of the cells, as well as some of the viruses can cause apoptosis. Some virus particle can be expressed over the cell surfaces by MSC1 that can be sensed by these T cells, and they can kill these infected cells. So once these virus infect that particular host and spread locally as well as systematically, it leads to production of some of the deleterious effects. It can be cytopathic or non-cytopathic. It can modulate the immune system and can produce hypersensitivity reactions or autoimmune diseases. Also, it can cause the sublethal genetic injuries and can transform the cells into the neoplastic ones. So first we will discuss about the cytopathic changes. Cytopathic viral infections ultimately kills the cell in which they replicate by preventing synthesis of host macromolecules or by producing degradative enzymes or toxic products or by inducing the apoptosis. Virus-induced metabolic and toxic insults to the cells may result in morphological changes indicative of cell degeneration and necrosis. Virus multiplication in the nucleus and in the cytoplasm can lead to the formation of inclusion bodies in cytoplasm or in the nucleus. Virus can inhibit the host cell protein productions, and that can lead to the cellular degeneration and necrosis. Virus can also interfere with the cellular membrane functions. It can increase the permeability and membrane potential so that there will be disruption in the fluid and ion balance that can lead to the disruption and lysis of these cells. At the same time, envelope virus can insert their surface glycoprotein into the host cell membrane, and it may lead to membrane fusion between infected and non-infected cells that subsequently leads to the formation of syncytia, that is multinucleated cells can be seen in the infected organs. Some of the virus can disrupt the cytoskeleton. As you all know, cytoskeleton is very important for the maintenance of rigidity and shape of the cells, as well as it maintains the motility of the subcellular structures like mitochondria, Golgi apparatus. So loss of these cytoskeleton like microfilaments, intermediate filaments and microtubules lead to the changes in the shape, and it can produce the rounding of the affected cells. Also, some of the viruses can produce the apoptosis by activation of intrinsic or extrinsic mechanism. In intrinsic one, it can cause the alteration in the mitochondrial cell membrane that leaks the cytochrome C into the cytoplasm, and then it will activate the caspases and ultimately leads to the apoptosis. Some of the non-cytopathic effect produced by the virus, it includes, actually non-cytopathic viral infections do not kill the host cell in which they replicate. On the contrary, infected cells keep on producing and releasing of virus. So such infection called as persistent productive infections, and in some infections it won't produce the virus progeny, that is called as persistent non-productive infections. Here what happens, it produces some of the proteins which helps to maintain the homeostasis, and it minimally affects the such host cell protein productions. Such type of infection generally produced by the RNA viruses like pestiviruses, adenoviruses or retroviruses. Some of the persistently infected virus may go into latency. Latency is a form of persistent infection in which infectious virus is not demonstrable except when reactivation occurs due to immunosuppression. It maintains by restricted expression of genes that have capacity to kill the cell and express only a few genes that are necessary for maintenance of latency of that virus, that are also called as latency associated transcripts. Some persistently infected cells by the viruses may lead to these non-lethal genetic injuries that may progress these cells into the indefinite multiplication without any control, and subsequently that leads to the cancer formation or neoplasia. Some of the viruses like retroviruses, herpes viruses, papillomaviruses can cause such tumors in the affected cells, as well as virus can modulate the immune system and can produce the immunopathology and elicit the hypersensitivity reactions like antigen antibody complexes may form and can deposit in the blood vessels as well as in the glomeruli, that may lead to the immune complex mediated glomerulonephritis and vasculitis. Like in case of feline leukemia virus or equine infectious anemia virus, generally these type of hypersensitivity reactions seen, and it produce the typical lesions and signs in affected animals, as well as it can produce the autoimmune diseases. Autoimmune disease, it is just a molecular mimicry phenomena wherein virus has got the epitope which has exactly similar structure with the host cell, like in myelin basic proteins. So when such virus affects such host, there will be formation of antibodies against such epitopes, and these form antibodies can destruct the host epitopes as well, and it leads to the production of autoimmune diseases. So these are the some of the mechanism by which virus causes damage to the host cells. So these are the some of the references from where this material has been compiled. So thank you for watching this presentation and video. Thank you.